Aloha, my name is Sheina Sim, and here I will be presenting some of our work on the Queensland Longhorn beetle. I am a research biologist at the USDA Agricultural Research Service, and we are one of almost 100 locations that represents the research arm of the USDA. This is a photo of our research center, and it's located just Malka of UH Hilo campus in the Science and Technology Park on Novello Street. In our lab, we do applied research that is focused on insect pests of tropical crops and commodities. At the USDA, I primarily study true fruit flies, which are major pests of fruit and specialty crops. And my area of focus is fruit fly genetics and using genetics to better understand how to control, suppress, and eradicate fruit fly populations on a large scale. But I'm not here to talk about fruit flies today. We're here to discuss this beetle. We've been calling it the Queensland Longhorn beetle, and the reason it is a problem is because it is known to attack cacao, citrus, breadfruit, and other trees that, though they're not necessarily fruit commodities, they're ones that we care about, such as the kukui. The Queensland Longhorn beetle, or QLB, belongs to a family of beetles called Cerambycidae, or the Cerambicids. These are the longhorns. Other notable members that are causing problems to mainland trees are the Asian longhorn beetle, which attacks hardwood trees such as maples, and the citrus longhorn beetle, which, as its name suggests, attacks citrus trees. Because they are insects, they are ectothermic, which means that their biological functions, such as growth and activity, are dependent on their environmental conditions, such as temperature. Their long horns aren't horns at all, but they're actually long antennae, which they use to interact with the chemical world. The adult beetles themselves aren't the ones that are causing damage, but it's their larvae which eat wood. This means two things. One, that they can move around as larvae without being detected, which makes them potentially very invasive species. But also, since they eat wood and can destroy living trees, they can be major pests in an area. Beetles are insects that go through a complete life cycle, which means that they begin as adults. They land on a tree, lay eggs. Those eggs hatch into early instar larvae. Those larvae grow to be mid-instar larvae, then late instar larvae, until finally it pupates into a tenural adult. That tenural adult then emerges into an adult and it starts the life cycle all over again. Because most of its life occurs inside of a tree, it is mostly invisible to us, which makes it difficult to detect and easy to move around without knowing. It's through this movement of wood that it likely came to us from its native range in Queensland, Australia, where it gets its name from. Prior to detection on the Big Island, it was really only found in these two places in Queensland and New South Wales. QLB was first detected in Hawaii Island in 2009, and between 2009 and 2013, there were no documented reports until multiple adults were found in Puna. These adults were found, but we didn't know yet what it was eating until 2014 when larvae were found in an infested tree. In 2018, it was then identified to be causing problems in two local cacao growers, and the Hawaii Department of Agriculture and the Big Island Invasive Species Committee declared it a pest in Hawaii. Coincidentally, in 2018 in September, I also started my current position with the USDA, and though my expertise is in fruit flies, it was apparent to me that investigating this beetle to try to develop ways to manage it and minimize its damage was important. Since then, I've procured some outside funding through the Hawaii Invasive Species Council and used that to hire some help to do our research. At this point, we've started several projects looking at their attraction to lures, their geographic distribution on Hawaii Island, and population genetic analysis to answer questions about the nature of their invasion, which takes us to present day. QLB in Hawaii has several confirmed hosts, such as the breadfruit, cacao, several species of citrus, and kukui. It also infests many other trees. And though the infestation of cut logs doesn't necessarily mean it can infest live Norfolk pine or avocado, and thus are a threat to those species, cut logs can be infested and can represent pathways by which QLB could be moved around, for example, to other parts of the island. Some signs of damage to infested trees include abnormal oozing that can become apparent when sap comes from the hole chewed by the mom to lay eggs and represents the unimmune response of the tree, girdling, which can kill tree limbs, and of course these wood shavings and exit holes, which signifies adult beetle emergence. Because of my background in population genetic analysis, I wanted to use molecular methods to investigate the nature of its invasion to this island so that we can determine how it got here and use that information to prevent future invasions. We know that QLB is native to Australia, but did it arrive on the Big Island directly, or was there an intermediate source that got it from Queensland beforehand? And wherever it did come from, did it invade just the one time, or did it come multiple times? Once it got here, how much is it moving? Does it arrive, emerge as an adult, and then fly as far away as possible? Or does it mostly stick around from the tree that it came from? What our population genetic analysis has shown us so far is that there's no genetic diversity of QLB or in QLB at the genetic marker that we investigated. This means that there's evidence that the introduction of beetles from outside Hawaii has only happened one time. Because there's no genetic diversity, we could not use that to investigate dispersal. So we developed a reporting tool so that anyone who found a beetle could report that detection and send a photo and tell us where beetles are. We received reports from all over the island for these beetles, but when you look at the photos, the only ones that were QLB were all from this area in East Hawaii. Each of those dots represents a report, and I use the photo they sent to identify the beetle reports to species. If you zoom in to where all the beetle QLB were, you see that they're mostly clustered in the Hawaii Paradise Park, Orchid Land, and Keau area, with these two reports in Hilo, one in Panaeva and one off rail road close to the Hilo transfer station and green waste facility. This tool has been incredibly useful at tracking the spread of the beetle because until these reports, we weren't looking for QLB in Hilo, but now we know that we should. The mapping reveals that the dispersal of the species is actually quite low and likely mostly human mediated. In terms of mapping, trapping, and lure development, one thing that we learn in pursuit of developing a trap is that lures are not very effective, at least not the ones that are commercially available. So they're not effective for control, nor are they effective for early detection. This is disappointing, but not surprising because lures are designed to be very species specific. And since this beetle is very new in terms of being a pest, not a lot of work has been done on it. There's only been one study on QLB which looked at their attraction to several commercially available lures, and based on 2 trapping experiments which tested their attraction to pheromones, one experiment showed that the pheromones were a deterrent because they caught fewer beetles than the control, although this is not a statistically significant result. And the second experiment showed that the treatments were all equally bad at not attracting beetles. Some potential methods for beetle control include systemic insecticides, such as imidacloprid soil drenches. Unfortunately, these have not yet been tested in Hawaii and may be very expensive to do on Hawaii Island. This particular pesticide is also not particularly good for pollinators. Another potential method for control is Bavaria bassiana, and this is a biological pesticide that uses an insect-infecting fungus which can kill an adult or larva. This is used on the Big Island currently to control coffee berry borer, but is not labeled for QLB and thus would represent an off-labeled use of the product. Some anecdotal evidence of its efficacy are these two beetles in an adult on the left and the larva on the right, both of which are infected with fungus. More research needs to be pursued to determine if the fungus is indeed Bavaria, but the white fungus on the joints of the adult beetle on the left are characteristic of Bavaria infection. So while we do have a lot of work to do, the current best recommendation is to survey, sanitize, and report beetle infestations. The life cycle of the beetle is very long compared to other insects such as fruit flies, and they live for a few weeks to months within the wood. Since trees exhibit signs of infestation, it's possible to catch newly infested trees before too much damage has occurred. Once damage is detected, action can then be taken to remove the infested wood and destroy it. My lab is currently also pursuing other QLB projects, one of which is determining beetle infestation from larvae and frass samples. Since there are many species of longhorned beetles on this island, it's difficult to tell their species apart based on larval morphology alone, since the immature stages of the different species look very similar. Because we're developing methods to do genetic analysis, we can apply this to the larva so that way we don't have to wait for it to emerge as an adult to know what species it is. Additionally, we will also want to be able to develop methods for genotyping frass to determine if frass from on a tree is actually QLB or from another species, in which case you wouldn't need a beetle sample at all to do that test. We're also in the process of establishing an experimental research colony that we can use for lure development and also testing insecticides. Hopefully this colony will enable us to provide recommendations for lures that can be used to trap and detect beetles, and also specific pesticides that are effective at controlling adult beetles. Here's one of our team members, Sheina, who is making sure that the beetles are being taken care of in times of COVID. So if you suspect that you have beetles, if you have a live adult, you can bring it to us and we can either use it to supplement our colony, or if it's dead, we could use it still for genetic analysis. If you bring us material, we would like some data, such as where it was found, when it was found, and also what hosts are near it or in the area where it was found. If it's a larva, then you'll know what tree it came out of. If you point your phone at this QR code, it'll take you to this website. The URL is down here on the left. And you can use this to complete the report form on your phone, and then also use it to submit a photo of your beetle capture. And then once you've done this, that beetle will be represented in that map that I showed you. If you can, please bring the beetle here where we can receive and process it. The address is in the top left. If you can bring it in a glass or hard plastic jar, that's best, because it can chew through cardboard and soft plastic, for example, like a plastic bag. And because it can do that, if you try to transfer it or transport it in a plastic bag, there's a high likelihood it'll get away. So with that, I would like to thank and acknowledge all of my coworkers and collaborators. All this work could not have been done if it weren't for the hard work of the people at PBARC, and also my collaborators at the Big Island Invasive Species Committee, HDOA, and USGS. I'd also like to acknowledge the East Hawaii Cacao Growers Association and the Hawaii Chocolate and Cacao Association, who have growers who have graciously permitted us to trap for beetles on their property. Finally, I would like to thank the organizations who have funded this work, the Hawaii Invasive Species Council, and internal funding from the USDA. And lastly, I would like to thank you all for listening.